जैसा कि हमारे पास अब टेम्पलेट स्ट्रैंड मौजूद है टू परफॉर्म द सीक्वेंसिंग रिएक्शन, then we now proceed further to synthesize the complementary strand of the template. For that purpose हमारे पास 3 different methods हैं जिनमें से हम किसी एक को भी चूज करके आगे चल सकते हैं, and these 3 methods differ from each other in the choice of enzyme. Like we have the choice of enzyme Taq DNA polymerase, we can use Klenow fragment, or we can use Sequenase enzyme to synthesize the new strand which can be sequenced later. DNA polymerase is preferred over Sequenase or Klenow fragment because this is a conventional enzyme which can catalyze the incorporation of dideoxynucleotides in place of normal ddNTPs in this method of sequencing. That means कि जो हमारे dideoxynucleotides हैं, they can equally be treated like normal nucleotides, and polymerase can incorporate them into the newly synthesized strand. DNA polymerase, the Taq polymerase, reduces the problem of mismatching of primers to anneal. Sometimes what happens कि हमारे पास जो gene specific primers हैं, if we use other enzymes, they can mismatch and they can synthesize unwanted or unspecific fragment of DNA. Further, DNA polymerase helps to sequence those templates of DNA which have high degree of secondary structure. So if we use Taq polymerase, then those DNAs which have the secondary structure, they can also be sequenced with good efficiency. At the same time, there are some disadvantages of DNA polymerase, like it catalyzes the incorporation of dideoxynucleotide triphosphates at a slow rate. Polymerization of ddNTPs is slow as compared to the polymerization of normal ddNTPs. Then there is visible band-to-band variations. The fading of bands is visible from bottom to top. Bottom to top means when we do the gel electrophoresis, जो bands bottom पे आए हैं, they are having shorter size as compared to those which are at the top of the gel. They are longer in size. So the longer is the DNA fragment, it is going to be faded in the electrophoresis, and sometimes shadow bands appear. This could be due to the reason that Taq polymerase has 2 activities. It has proofreading ability as well as it has polymerization ability. Scientist has resolved these disadvantages or problems by using an improved Taq polymerase, and that improved Taq polymerase has only the polymerization ability. Its proofreading ability is removed, and ultimately that efficiency of incorporation of dideoxynucleotide is increased by this modified Taq. Now, mutated Tags are also made available in the market for use in place of the wild type enzyme, which exhibit reduced discrimination between these 2 types of nucleotides, that is dideoxy and normal deoxy. So these mutated enzymes don't consider them dideoxy or deoxy, they incorporate them almost at the same rate. So now we have to talk about the labeling mixture. We have the template DNA, we have the dideoxynucleotides, we have the normal deoxynucleotides. We choose the enzyme if we are going to work with the mutated Taq, normal Taq, or we are going to work with any other enzyme. We have to decide the concentrations of dideoxy and deoxynucleotide phosphates. Like for example, we use normal nucleotides at a concentration of 150 micromolar, and we are going to use dideoxy at a concentration of 1.5 micromolar because they are going to be incorporated at the less ratio as compared to the normal one.